expect a follow-up email that will have both a link to the recording of this section and also a link and reference to, for instance, the Jupyter notebook that you're looking at now and some of the other documents that Dr. Sen will be presenting to us today. We started this seminar series as a way to almost replace or go to a pandemic version of what we usually do as a summer workshop that goes over quantitative genetic tools for the rat, but obviously we had to cancel it this year, and we were hoping that this might even be able to hit a much bigger audience when we're not asking you to come join us for a day and a half. The way we're going to run this, because we have so many participants this time, is that we will do questions over chat. So I will be monitoring the chat board. So as we go along, as questions come up, I'll pass those on to Shauna, because I don't know if you've done a Zoom presentation before, it's kind of hard to see your entire screen and monitor and talk in the chat at the whole time. So I'll be taking that responsibility. The next webinar will be again in 2 weeks, and this Dr. Rob Williams will be taking over that one, and he will give us the next step in our rung. So he'll take us from QTLs to gene variants. If you did, one more general announcement too, is if you did register for this and log on, just because I'm trying to learn my Zoom as best I can, if you registered within the last 30 minutes, can you drop me an email with your email address if you would like to continue to get announcements about this webinar series? I think that's all that I had as far as general business. Did you have anything you wanted to add, Rob?
No, I'm good. Thank you.
I think I forgot to formally introduce the group. That I am Dr. Laura Saba. I am one of the co-directors of the NIDA Center of Excellence in System Genetics, Omics, and the Dictome. And the other director of that is Rob Williams. And Shauna, who's going to present in a minute, is lead of one of our cores within that center. So with that, I'll pass it on over to Shauna. And like I said, I'll be monitoring that chat board. If you want to push questions that way, we can get those to Shauna, or we can get them answered one way or another. And everybody, when you joined, I'm sorry, let me add one more note. You're automatically on mute. If you take yourself off and we start getting background noise, I will go ahead and mute you again, just to try to make this as smooth as possible for the entire presentation. All right, I'll hand it over to Shauna then.
Thank you, Laura. And thank you, Rob, for this opportunity to talk about and introduce quantitative genetics mapping to all of you. This is all very new to me, doing a Zoom webinar, so I hope you will apologize if there are any glitches. And please do give us feedback what you might have changed, because we hope to do similar things in the future. So your feedback is going to be very useful. The other thing I should say is that this is going to be a very elementary talk, at least from my point of view. And my main goal here today is to explain the intuition and the basic principles behind genetic mapping, especially for quantitative traits. There are many illustrious individuals in the audience. I've looked at the registration list and I can see the list of participants. There are a lot of people who know more than I do in many things and have much more nuanced views about a lot of things. So I want to make that clear. If things are too simplistic or not subtle enough, we hope that we can have a discussion in the future. The other thing I wanted to add is the lecture I'm going to give you is basically a Jupyter notebook. So everything, and both of these have been posted onto the website, which I think Laura will share or has shared. So you can look at it and tweak it, change certain things if you're interested. And I'll be using two notebooks. One is the slides that you're seeing, and the other is a slightly more in-depth notebook on kind of a basic analysis of a rat data set that Dr. Leah Solberg and colleagues performed over a series of studies. So you can look at both of those. The downside of the Jupyter notebook is that the view that you see is maybe not what you're expecting, like a PowerPoint slide. So, but I hope it's useful in its own way. So here are my goals. The goals are to basically define what is a quantitative trait locus, why are we interested in them, what are genome scans, and how do they help us find QTL, and what are the strengths and limitations of this approach. So these are the broad goals, and the other lectures in the series will follow up on this and talk about in greater detail, what you do afterwards, how can you improve it, and other aspects of finding or using genetics to find or dissect the etiology of a trait or a disease. So before we talk about what a QTL, quantitative trait locus, is and why we're interested in, I think we can have maybe a quick overview of why we are using rodent models at all. And this may not be news to most people in the audience, but just as a, maybe to reiterate, rats and rodents in general are mammals and share a lot of the DNA with us, including, and also a lot of physiological processes are common between humans and rodents, even though they might look different or have a very different shape or size. Many strains mimic human phenotypes and therefore are useful as models. On the other side, the reason they have a short life cycle, small size, they are suitable for genetic manipulations, and so are convenient as models, with the understanding that they are not exactly capturing the human condition, but getting somewhat close, or getting the basic idea is likely similar. There are a lot of genetic and genomic resources, such as strains, outbred populations, sequences. All those things make it very convenient to study rodents. And finally, some studies or interventions are not feasible or ethical in humans. The only way to study it would be in some kind of model system, and at least a little bit removed from humans to be ethical. So what is a quantitative trait? So quantitative trait is, anything that you can measure is a quantitative trait. Even something that you might think is qualitative, such as coat color, you could treat as quantitative. If you color it, is it black or white? So while the term quantitative trait appears over and over again, it really means anything that you could measure. And it doesn't have to be a single number. It could be a whole collection of numbers, such as a growth curve or the activity pattern in a mouse or rat in a cage system. So it could be like multivariate or multidimensional as well. But I'm just using quantitative trait as a shortcut for all of that. So the anchor data set that will run through today's lecture is an F2 cross between the F33 Fisher F344 rat and the WKI rat strain, an F2 cross between the two. This was done by Leah Solberg and colleagues in a series of studies that were conducted to examine the genetic connections between behavior and physiology, mainly depression, but also other things like the HPA axis. So it's a number of papers, and I'm giving you the citation here at the bottom of the last paper in the series that maybe there are more, but last that are in the database where I pulled the data from, which is the QTL archive. And below here is a little bit of code in R that I use to enter the data. You don't need to worry about it right now, but and the images here are of the two rat strains. I mean, to me, they look very similar. They're taken from Charles River, so if I messed up, please forgive me. But this is my understanding what the two rat strains look like. But physiologically, the WKY rat on the right has a lot of physiological and behavioral similarities to human depression, which is why it was chosen as a model for this cross. And the other strain is a genetic, kind of the quote-unquote control strain, the F. Fisher 344 strain. So that's the background of the study. And here is a little histogram of one particular phenotype, which is glucose that was measured after a stress, after the animal was restrained. And so although glucose is a metabolic trait, in this context, it's you could think of it as a behavioral trait because it's reflecting the animal's response to the restraint. So you can see there's a wide range of variation in the animals in this cross. If you look a little bit closer in using this so-called violin plot. So this is just like a histogram, but kind of turned 90 degrees. So now the glucose levels are on the left-hand side in the usual milligrams per, I think, deciliter. Let's see. Yes. And you can see very clearly a difference between males and females. So this is, again, and second, in the salmon and blue colors, I've annotated the average levels of the WKI in blue and the F3344, the Fisher, in the salmon color. So a few different things in this figure. First, males and females overall in the F2 population have different levels. Number two, the two strains, the parental strains from which these animals were derived, have very different glucose levels. The WKI strain having higher levels at both strains, although the difference between the strains does not seem to vary by sex. They are different. And the third thing is that you can see that the range of variation in the F2 animals is actually quite large and exceeds the range that you see in the parental strains. This is not entirely obvious here from this figure because you don't know the slack around this, but and this is also very typical of almost any genetic cross, that the range of phenotypic variation that you will see will typically exceed the range of phenotypic variation that you see in any of the parental strains. So now I'll show you another little cross. This is a mouse cross between two mouse strains, but it was a back cross instead of a intercross in rats. And here it was also a behavior study where mice were followed in cages, and the chance that they are active at a given time of the day is plotted here. It's called the active state probability. And it's a number between 0 and 1, and you can see that there's quite a range here. The reason I'm bringing this up is because there are some things are just a little bit easier to see in this back cross than in the F2 cross, which is quite, very rich, but it has some complications. So I'll just sometimes I will switch to this data set just for clarity purposes. Okay, so going back to, why are we interested in in QTL? So I think this will be a kind of philosophical thread, but I think is central to the reason why experimental crosses and QTLs are so interesting. So almost all of science that we use today have roots in, roots that go long back, but the methods that we use for causation are broadly derived from the British empiric tradition, and two contemporaries, John Stuart Mill and R.A. Fisher. And the two methods that we use are, first, it's called the method of difference. This is from John Stuart Mill. And the idea is that if a phenotype is observed under identical experimental conditions in say, a congenic strain of mouse, but not in the background strain, then the genetic variance in the congenic regions are the cause or a necessary part of the cause of the phenotype. So here the idea is that there are two experimental units that are identical in every way except for one thing. So if we see a difference in the outcome, it must be due to the one thing that is different. So, congenic mouse strains, a lot of basic science is done in this way. You keep everything constant except and vary one thing. If you see a difference, it must be attributed to the one thing that you changed. So this is, the approach here is to eliminate all variation except the cause of interest. So the other piece, the other way of establishing causation is randomization. And the classic thing is randomized clinical trials. Here, the treatment is randomized to subjects. Subjects are observed, and if they have different outcomes compared to control subjects, then the treatment must be the cause for the different outcomes. So this is used in humans in clinical trials. Here, the idea is a little bit different. Here, you are embracing variation. When you do a clinical trial, everybody is different. You cannot eliminate variation. And randomization is a way to get around that. By the act of randomization, you can determine cause in the presence of variation. And in fact, this will have a little more generalizability than what you might do from the method of difference, because there you are fixing everything except one thing. Here, you are embracing the variation that exists and then randomizing an intervention. So, they have all pros and cons. So where do experimental crosses fit in? So the idea behind experimental crosses is that, if you have two inbred strains, such as the WKI and Fisher strains, that show consistent difference in the phenotype in the same environment, then we know that the difference must be genetic. So this is the method of difference. Now the question is, how do I find that? What is it that is causing the change? So the idea of experimental crosses is that you create a family by mating the two strains, and that creates a population of approximately randomly shuffled genomes. So this is now like a randomized trial. in that the genetic constitution of the individual is being randomized in the population. And therefore, associations between phenotypes and genotypes may be interpreted to be causal. This is very important, and this is what allows us to infer causality from an experimental cross. So here is just a schematic of what might happen in a F2 intercross. You have these two strains, the pink and the purple strain. You cross them. They're genetic. They are two inbred strains, so they're both chromosomes are identical. In the F1 generation, individuals have one chromosome from each strain, and in the F2 generation, these chromosomes recombine to create a mosaic that is approximately randomized by meiosis, and you get a mixture of this pink and purple. This is what happens with a F2 cross from two strains. In something like a heterogeneous stock, you might have a bunch of strains that might have a complex mating scheme, and then you end up with a more complex mosaic. But the end result is the same in that you get a collection of approximately randomized genomes, and therefore associations that you find in these populations are likely to be causal. And now I'm going to show you, make a causal diagram to underlie kind of what is the kind of the principle underlying what we are trying to do here. So what is, and if there are epidemiologists in the audience, I think they might be able to relate to this. So what is happening is the following. Meiosis is randomizing both the cause, the QTL, the quantitative trait locus, as well as all the markers that we can observe that tell us about the genetic constitution of an individual. The QTL is not observed directly. We don't know where it is. We can see the marker. We can see the phenotype. And because meiosis is the common agent that is both randomizing the QTL and every marker that you have on the genome, markers that are near the QTL will show an apparent association with the phenotype. Okay, so the facts are what this causal diagram is telling you. Meiosis randomizes the observed QTL and the observed marker. These are these two arrows. The QTL causes the phenotype. QTL causes the phenotype, straight arrow. And this leads us to observe an apparent association between marker and phenotype. So these are the dotted lines. These are the associations caused by this common meiosis that is common to both the QTL and the marker. Since the QTL is not directly observable, we test for association between genetic markers and that are observable and the phenotype. Markers associated with the trait are likely to be physically close to the QTL. So this is the basic idea of the genome scan and what we are doing in any kind of QTL analysis. So now I'll show you a few variations of this causal diagram, and the reason for showing is that it's important to keep things clear. That sometimes it is not clear what is a covariate, what you should adjust for, what you should not adjust for. So this is now a causal diagram for what I would call a covariate. So it is very similar to the previous one, except now we have an extra factor like a covariate. So in this Leah Solberg's cross, a covariate would be something like sex or cross direction or something like that. So here the important thing is that the meiosis is randomizing the QTL and the marker just like before, which leads us to observe an apparent association between marker and phenotype. In addition, there is a covariate that contributes to the phenotype, such as sex, as we saw. The covariate sex does lead to a difference in the phenotype. But the covariate is causally unconnected to the QTL. So here I would add a caveat. By here I mean not on the X chromosome or Y chromosome. Not if the QTL is not on an autosome, then it is not directly causally connected to the QTL. So this is what I would call a covariate. There is no direct connection between the QTL and the covariate. And the QTL contributes to the phenotype directly and not through the QTL. But there are cases where sex could interact with the QTL, in which case this diagram would not hold. So, but in many cases, something like sex, or it could be things that you vary would be in this time.
Shanak,
So,
Shanak, we
yes.
have a question from the audience that would fit well here. So does randomization establish causality or association because all factors that differ are not known or cannot be measured? Same with the differences. Isn't an assumption made that they are identical except for the factor being tested? For example, microbiome, parental effects, other subtle microenvironmental differences. But the test for differences in randomization are powerful. But I would think it would be hard to claim causality unless one could show that the underlying assumptions are true or reasonably true.
Yes, that is correct. So I think that, I think I would not disagree with the overall statement. I think that, okay, so here we are not actually randomizing. And this is a longer discussion. We have a set of approximately randomized genomes, but the randomization of the genome is coming before the phenotype. And so I think things of the example like microbiome, those things, the microbiome is what I would call a mediator, which is what is coming next. It is something that is connected, could be affected by the QTL of interest, and could also affect a phenotype. And in that case, it is not entirely clear whether you should adjust for the mediator or not, or do some other analysis. The other piece is that whenever the beauty of randomization is that you don't need to observe everything that is potentially causing potentially causing the phenotype, as long as the process of randomization is not affected by those factors. So for example, in QTLs, one way that might be affected, if I was doing any kind of selection based on the phenotype, then this diagram does not hold. So there is a lot of subtlety involved, and I would agree with the essential sentiment, but I would say that the randomization of genomes that occurs during the process of meiosis gives us a powerful tool that something like a congenic, would not. Well, okay. Which is qualitatively different from the type of evidence that you would get from, say, something like that uses a method of difference. So. Okay. So this is what I wanted to show is potentially what is the difference between a mediator and a covariate. So these are things that I think is important to think about, and this is related to the question earlier, is that at some level, the analyses that we do in genetics are very simple, at least technically simple. It is still very important to think about, like, what are we testing, what are the roles of the different data that we are using to conduct this analysis, and what are the assumptions underlying it. And some of those assumptions, as pointed out by Dr. Nadeau, are not testable. So that was one of the reasons why I wanted to put up these diagrams. Okay, so what does what do the genotypes, marker genotypes in a cross looks like? So this is a F2 intercross. Now I'm showing a mouse intercross. This is how the genotypes in a mouse cross, back cross would look like. Here, every row is an animal, and the two colors represent the two parental genotypes, red and the blue. What you can immediately see is two things. If you look vertically, every vertically each line vertically is a specific point on the genome, and roughly speaking, is equally red and blue. So if I look at any given point, it is like a coin toss. The genotype is a coin toss, like randomized, like a clinical trial, red on blue. But if I look across, you will see that nearby locations have a similar color. So this is a kind of so it means that if it is red here, it is more likely to be red at a nearby location. So this is like a type of genetic confounding. So this is the pro and the con of the randomization that the approximate randomization that we see in an experimental cross. So the picture for the Solberg dataset is a little more complicated because there are 3 genotypes and there are these dominant genotypes as well, and so it is less clear how the genetic randomization is working, but here it is if you are interested in knowing what that looks like in the Solberg cross. Now, if you look at the marker genotype, the actual marker genotypes, again, I'm going to the mouse data set, the back cross, you can see how the data is actually on a given chromosome. I've selected just about 20 individuals. So what you see, each line, each horizontal line is an individual, and each like vertical, each bead is like a genetic marker at a given location in a given individual. So these are all the same marker in different individuals. So this is another way to visualize the genetics. The thing that it makes clear is that the marker locations are not uniformly spaced along the genome. Here, the distance is actually on the genetic scale, genetic location, not physical location. And you can see that there are gaps in our information about the genetic constitution of an individual. And this is where some of the statistical approaches come in. But at the end of the day, what you're trying to do is you're trying to infer what's happening in between 2 points where you don't have a marker genotype. So if you have a region of the genome that is tightly genotyped and they are have the same genotype, you can be very confident that this is white here, one strain. If you have a region where you have an individual with 2 markers with different genotypes, then you know that their crossover occurred somewhere in between and you don't know, and there is a little bit of ambiguity there. So this is the same figure, but for an F2 cross. Here the beads are, the black and the white are homozygous AA and BB. If you have, and the grays are the heterozygous. And you can see the same pattern. Nearby locations have similar genotypes, and sometimes they switch the genotype. Okay. So going back to, so what is a QTL? It's a region of the genome, a locus that contributes to variation in a quantitative trait. Experimental crosses provide strong evidence. I would not say they determine or rule out everything, but they provide strong evidence of causation. And finding a QTL is usually the first step in dissecting the cause of that trait variation. And so how do we find QTL? Again, to reiterate, since a QTL is not directly observable, we test the association between genetic markers and the phenotype, and the markers that are associated with the trait are likely to be physically close to the QTL. This is basically the idea of the genome scan. And now for the next maybe 10-15 minutes, I will switch to my other notebook with the analysis of the rat intercross. And here you will see a little bit of the nitty-gritty of how these analyses are done. I will not explain the code, but I will explain the logic and show you kind of the workflow of how it works. First, I read in the data. I had to tell the RQTL program how to interpret the code. The neat thing is that things were pretty well organized, and except for some warning messages, I did not have to do much. Here is a summary of the data. There were 25 phenotypes, including sex, cross direction, and litter. There are a lot of details here that I'll ignore. There's a small percentage of dominant markers, but otherwise the genotypes are in the expected 1 to 2 to 1 ratio. You can see that in the autosomes, about 25% are homozygous F344, about 50% are heterozygous, and about 25% are homozygous WKY. I think I'm seeing some questions in the chat. Can I answer them later? I think,
yeah,
if you don't mind, but I will get to them.
Yes, I've got them marked.
Yeah. So here are the phenotypes. There are a bunch of phenotypes. I'll just focus on one particular phenotype, which is the glucose 30, which is the glucose after the animals were placed in a restraint tube for 30 minutes, and blood was collected after that. So again, it's a metabolic phenotype, but with a behavioral interpretation. So here is what the these are. Here's the glucose, and you can see the variation in the glucose levels, and So first thing, what you have to do is you have to think like any other data analysis. So doing QTL analysis is not any different from a general data analysis, except that you have some additional considerations that relate to the genetics. So when you see a trait like this, it's a quantitative trait, as you would expect. But like a lot of positive traits that are inherently positive, it often benefits from a log transformation. And so if you do the log transformation here, you can see that it makes it a little more symmetric. It is not essential that you do something like this, but it is often helpful and might improve your statistical power a little bit. So it's something useful to do. So you can also do scatter plots with different phenotypes. So here there is another phenotype, which is glucose challenge that was done 2 weeks later, glucose challenge test. And you can see that there is a correlation between the 15-week glucose levels after the restraint and the post-glucose challenge test. If you transform the log, it looks a little bit better. So Based on this, we decided to analyze the traits on the log scale, as did the study authors. So let's also compare by sex. So again, this is the Violin plot. I think you've seen this before. There's a difference between by sex. The glucose challenge also shows the same thing. Now let's explore the genotypes. I showed you the genotypes a little bit. Here is the genetic map. So the genetic map is like the physical map in that it shows you the approximate locations of the genetic markers that were studied in this cross, with the difference that the scale of the genetic map is a little different from the physical map. The physical map has to do with the base pair position of a particular marker. The genetic map has to do with with the density of recombinations in it is rescaled with respect to the density of recombinations in any given interval. So if a physical interval of like 10 megabases has twice as many recombinations as another 10 megabase interval, then the genetic distance in the first one will be larger than in the for the second interval. So that's so here you can see that the genetic markers are on whole, I think, about 20 centimorgans apart. This study was done like almost 20 years ago, and that was state of the art, and it was a very good design. Now a modern study would probably have much more dense markers. But the 20 centimorgans, I think this is related to one of the questions. I think 20 centimorgans is more or less the minimum. Like you don't want to go any more than that. That is, you want your markers spaced at least, so at most 20 centimorgans apart, no more than that. So here are what the genotypes, again, a similar figure that I showed you, how they look on the genome, on chromosome 1, just to give you a visual of what they might look like. And next, I'm going to do a genome scan. So what is a genome scan? The genome scan is I'm walking through the genome, and I'm testing for association between markers at every position in the genome and the phenotype of interest. And if I do that and plot the result, this is what I get. On the y-axis, I have what's called the Lodge score, which is basically like an ANOVA that the larger the Lodge score, the stronger the evidence of association between genetic variation at that position in the genome and the phenotype of interest. So so this was popularized by Lander and Botstein in 1989, like almost 30 years ago, and was a big breakthrough in quantitative genetics. So as you can see that there is evidence that there are some places in the genome that appear to show high, like strong association between genotype and phenotype, at least based on a visual impression. So one of the questions is, how high is high? Like, where is our threshold for declaring that you would not expect such a high association by chance alone? Because so that's let's think about that a little bit. So what is the issue? So the main issue that we face with a figure like this is that you're doing multiple tests along the genome. To answer the question, the hypothesis that genetic variation is not associated with phenotypic variation, I'm doing hundreds, possibly thousands of tests along the genome that ask that question. So to answer one question, is genetic variation in this cross associated with phenotypic variation, I'm making 100 or 1,000 tests. So this would raise my false positive rate, because if I use many tests to answer a single hypothesis, I would have to adjust for the number of tests. And this is made a little more complicated by the fact that markers near are correlated, so it is as straightforward as the other situations where maybe you have independence and it's a little easier to adjust for multiple comparisons. So in QTL analysis, a common approach is to use permutation testing. And the idea behind permutation testing is that you shuffle the phenotypes with respect to the genotypes. So what you do is you're basically breaking the association between the phenotypes and the genotypes. So it is like the null hypothesis, while respecting the genetic structure that is observed in the cross and the distribution of the phenotype that is observed in the cross. So you are basically recreating data that have the same genetic and phenotypic structure as in the original cross, but you have synthetically broken the association between phenotype and genotype by randomizing the phenotypes with respect to the genotype. So if you do that, then the distribution of the maximum log score. So if you look at the peak log score in those reshuffled data sets, you will get something like this. So and contrast that with what you would get at from a chi-square distribution, this is the theoretical chi-square distribution, which has a distribution that is much to the left. So what does this mean? If I used a standard chi-square, then I would maybe cut. I think I may have made a mistake here. Sorry. Sorry, I had the scale wrong. So sorry, I apologize. I made a mistake. So, but the scale here is wrong. But the basic figure is correct. If you reshuffle the log score, this is the distribution of the log score. So you have to divide by about 5 to get the right number. So you would, so this 15 is more like a 3. So the permuted maximum log scores are about, if you want to see, this is the distribution of the maximum log score that you would get from the shuffle data set. And so things that are above 3, I'm just dividing mentally by, would be considered unusual. Contrast that with what you would have done if you just had a plain chi-square. If you had a plain chi-square, you might consider something like here to be unusual. So something under a log score of 1, you would consider unusual. So, sorry, not 1, like 1.5 yeah, about 1 point something it would be. So in other words, the distribution of the maximum log scores from doing a genome scan are going to be much higher than what you would expect from a chi-square. Fortunately, all of this can be done very automatically by the computer. And here are the thresholds that you would get from the permutation test. So here is the 5% threshold, which is about 3.5 in this. So this is saying that if I shuffle the phenotypes with respect to the genotypes, do genome scans, then 5% of the time I will get a log score that is above 3.5. 10% of the time I'll get a log score that is above 3.2. So if I add these thresholds to the previous genome scan plot, I will see that basically one locus exceeds this 5% threshold and the rest don't. If I have a slightly relaxed threshold, I get this dotted line, which is then maybe a second one. I get a second one, then maybe a third one. So now I can start thinking about adjusting for covariates. And here I will skip over the details, but basically, we saw that sex makes a difference. So I can repeat the genome scan with sex involved, with sex as an additive covariate. And you can see now that there are maybe the log scores are a little bit higher. They're in blue here when you adjust for sex, because it is picking up some of the variation, explaining some of the variation. And now if you do a permutation, now you have to do what's called a stratified permutation, is by permuting within sex, then you will get 3 QTL, so which is a little 2 more than what you would get originally. Now, if I go to these 3 specific loci, I can plot their effects, and here is the first one on chromosome 1, and you can see that this has a kind of a, well, I don't know what you will call it, dominant or recessive, but there's the WKY homozygous genotype has a higher glucose level, whereas the heterozygous and the homozygous Fisher don't. If I look at the chromosome 5 locus, this is what would be called an additive presentation. There's a continuous, the heterozygous is intermediate between the 2 homozygous. And the chromosome 20 locus is similar to the chromosome 1 locus, where the WKY homozygous locus has a higher phenotype, while the other 2 are about the same. So this is kind of your basic workflow of QTL analysis, which is similar to the general data analysis with some additional tweaks. One is you have to look at the genetic structure. I showed you, you have to do the adjustment due to permutation testing, and then you look at effects just like you would do for any other trait. So I'll switch back to the other presentation. So what are the strengths and limitations of QTL mapping? So the strength is that the approximately randomized genomes allow us to infer causal genetic elements, QTL. Infer, it is not a very cut and dry, but there may be some error associated with that inference. And we do this by scanning the genome for genetic markers associated with a trait of interest. So it's just like plain regression analysis, except that you are doing many regressions at the same time. So some things to keep in mind. So first is you have to choose a genetic cross depending on the constraints of your study. This is where your QTL design comes in. How many markers, what type of to choose, what type of cross to choose. These are some things you might have to decide. you have to exercise due care in data analysis. So examine the traits, covariates, mediators, investigating, handle outliers, just like you would do in any other data analysis. You have to examine the genetic markers for marker ordered errors, impute with, if needed, using EM or multiple imputation. These are some technical details I did not talk about that are kind of special to doing genetic analysis in these crosses. And you have to think carefully about the causal structure. What is the covariate? What is the mediator? So those are some things to. The genome scan is basically a linear regression that is multiplexed over all markers. So it is, in a way, it mimics what you're doing in the wet lab. The markers are a multiplexed assay, and the analysis is also a multiplexed analysis. And it has the effect that you have to make appropriate adjustments for multiple testings using permutations or stratified permutations. And in some crosses, like the heterogeneous stock, you may have to make adjustments for genetic relatedness, like linear mixed models and so on. The X chromosome, which I've glossed over, needs special care. I will leave it at that, and I'll point you to a reference. And the other piece I want to mention is that any selection based on the traits or the genetic characteristics will require adjustments to your analysis strategy, because it will kind of destroy the causal diagram that I showed you earlier. So the caveats are that the QTL will get you close, and close is in the eye of the beholder, but will typically not give you a gene. And you may have to leverage several lines of information to nail down a causal genetic element. And this is what this webinar series is about. And so if you tune in the future, this is what we will be talking about. And all QTL have a context. This is related to Dr. Neto's comments as well. The genetic background, the experimental conditions, whether it's a lab or a wild environment, and all those things will play into the interpretation and the robustness of the inference that you make. And here are some papers. This is the landmark paper, Lander and Botstein. This is Churchill and Dirge permutation testing. This is my paper with Gary Churchill, multiple imputation framework. This is the RQTL paper by Carl, which is the program I use for the genetic analysis. And the chromosome, X chromosome is just one chromosome, but it requires a lot of careful thinking. And I would strongly recommend that you read this paper. And finally, Gene Network, which will be featured in future talks and provides an interface for web-based genetics, which is one way for you to do some of the things that you have seen here.
Great. Shauna, can we get to, we're down to about 2 minutes. Can we get to some of the questions that were asked previously?
Okay, I'll go by.
And if I could maybe paraphrase and put a few together here.
Yes.
So I think there were a lot of questions between sex being a mediator versus a covariate, and how that, could you do a mediator within RQTL, and how do you make that decision?
So I think that it is not possible to do that. You have to do it, you have to tweak the analysis. It's not set up for mediation per se at this time. So you have to kind of tweak the tools that you have. So I would say sex is not a mediator, unless you are looking at the X chromosome. Then you can say that, because sex is caused by the genetics on the X chromosome. So on the X, you can say sex is the mediator, but for the autosomes, it is not a mediator. It is a covariate. But you have to keep those things very clear in your head, what the meaning of your analysis. So the short answer is, you need to think about the measure that you're looking at, is it a mediator or the covariate? Think about it like conceptually. And to actually do a mediation analysis, then you will have to tweak the existing programs. Okay, so.
And so that I think you answered the question about the density of markers, but I had another one from Joe
Yes.
that was sent to me privately. It
Yeah.
says, you nicely described the complication of linkage and tests for association, including permutation. Do we also need to worry about correlations among phenotypes? I.e., what if some of the phenotypes are also not independent? How is trait correlation taken into account?
Yeah, I mean, these are not easy. So, and I think that brings us a very great question. You know, I think this brings us back to this kind of diagram, like, here, what I'm calling a mediator here could be another phenotype or a trait, two correlated traits. They are it just it just depends on your thinking whether one trait is causing the other one, then it's a mediator. But if they are both causing each other, then they are like two multivariate phenotypes. And in that case, the best approach, in my opinion, would be to treat it as a multivariate phenotype. So in that case, the two phenotypes, this mediator vanishes, and you're back to this setup where the phenotype is multivariate. There are other questions like, what if they are not exactly the same thing, and there are different genetic elements that are causing one but not the other, and those are harder questions. This is at least the short version I can say. I apologize, I don't have a more thoughtful answer.
That's a whole other webinar.
Yes.
One of the next questions that I forgot to mention in sex from Milad was, how would you adjust for sex in the permutation test?
So one simple, so this is very well done I thoroughly recommend you read Carl Broman's 2006 papers. So one piece is to do a stratified permutation. There are three things you have to do for the X. One is you have to do a stratified permutation that is restricted to the sex and cross direction, which I have not talked about. The second thing is you need to adjust for the degrees of freedom of the test on the X chromosome, because it is different on the X compared to the autosomes. And the third piece is that the density of recombinations on the X is different, and is different in females versus males. And so you have to make an additional adjustment for the threshold. So there are three pieces. There is a stratification, there is a degrees of freedom, and there is a density of recombinations.
Shonak, one of the questions I saw that was interesting is, when do you have to use a linear mixed model as opposed to just a linear model?
So when you expect genetic relatedness between individuals, so in a F2 cross like this, the kinship between any pair of individuals is a half, and that is, they share about half their genome identically by descent. In practice, there will be some randomness around it. For more complex crosses, or even an advanced intercross, you will see patterns of variation that are where the genetic relatedness varies, is not exactly any pair of individuals. It is not a half, but maybe a little bit more than that for some individuals and less for others. And those are the situations where you have to adjust for the relatedness, and we could have a webinar on that itself.
Right. There was also a follow-up question about DO-QTL and RQTL2. And so just along those lines,
Yeah.
we are running out of time, but if you had a quick answer, but otherwise that would be a really good topic for another webinar.
Yeah, so DO-QTL and QTL2 are two programs for doing QTL analysis for multi-parental crosses. DO-QTL was designed specifically for the mouse DO population, and QTL2 is a general, will do almost everything that DO-QTL will do. It's written later by people who are connected, and is designed for multi-parental crosses and for high-dimensional phenotypes. So I would say most of the functionality of DO-QTL you will also find in QTL2.
By which we mean RQTL2, just I think.
Yeah, RQTL2, yes, that's right. And so I think if what I can do, I can respond to all these questions later individually. If we have the emails, I can, we'll have a chat transcript, right?
Uh-huh.
So I can take the transcript and answer everything and then post it or send it back to them. How's that?
That sounds like a great idea.
And for all of those of you who joined us today, again, in 2 weeks, we'll follow up. So if you have questions then, I think the first 5 or 10 minutes will be a reprise of some of the topics that Shaunak just went over in the context of Gene Network. We've put in the Solberg data into Gene Network. And if you're uncomfortable with R, you can do it in Gene Network. If you're comfortable with R, just work with the code and the Jupyter notebook that Shaunak has already sent, I hope, to most of you.
Yeah, and so you should have got a link to the Jupyter notebooks, but I will send a follow-up email to the whole for the webinar about some of the things that were asked for the repository, the video recording, a link to the next webinar, and a link to some QTL tutorials, and then finally that X chromosome reference that we'll send that one out again and highlight that for those that were interested. All right. Well, we are officially 8 minutes over. Thank you for joining us, everyone, and we'll be in contact soon.
Thank you.
Thank you very much. That was a great session.
Thanks, Shaunak.
You're welcome.